Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Srivastav. Today we are going to study the topic male sterility in plant breeding. So let's proceed. Let's understand what is sterility. Sterility refers an inability of a living organism to effect sexual reproduction. And what is male sterility? Male sterility is defined as an absence or non-function of pollen grains in plant or incapability of plants to produce or release functional pollen grains. In other way, male sterility is the failure of plants to produce functional anthers, pollen or male gametes, and male sterility generally occurs in bisexual plants. It was founded by J.K. Colruyter in 1763, who observed anther abortion within a species and species hybrids. Male sterility is more prevalent than female sterility. Male sterility is characterized by non-functional pollen grains, while female gametes function normally. Male sterility refers to a condition in which the plants are unable to produce or release the functional pollen grains as a result of failure of formation of development of pollen, stamens or gametes, which are male organs, as per Colruyter in 1763. And the main reason of male sterility is mutation. Now we will go through manifestations of male sterility, means events of male sterility. Male sterility represents absence or malformation of male organs, failure to develop normal microsporogenous tissue in anthers, abnormal microsporogenesis, deformed or inviable pollen. There will be abnormal pollen maturation, non-dehiscent anthers with viable pollen with sporophytic control, and there may be barriers other than incompatibility preventing pollen. Now we will go through the necessity of male sterility or role of male sterility. Because of male sterility, there will be reduction in cost of hybrid seed production. In hybrid seed production, we are using two lines, A line and R line, where A line, female line is male sterile line. Because of this, there will be aerial cross-pollination, which will eliminate the process of emasculation in hybrid seed production of many crops. It avoids enormous manual work of emasculation and pollination. In case of cotton or tomato or okra, we are not using male sterile line. There we have laborious work of emasculation process where we are removing the male pollens, and again we have to go for manual crossing. So in case of male sterility, if we are using these lines, male sterile lines, there will be no need of emasculation and manual pollination. Male sterility supports in production of large scale of F1 hybrid seed production, and we can speed up the hybridization program, and there will be commercial exploitation of hybrid vigor. History of male sterility. Colruyter in 1763 observed anther abortion or male sterility. Genetic male sterility has been reported in cabbage as per Renderfeld in 1960, and in cauliflower as per Hoff in 1961. Male sterility system have been also developed through genetic engineering as per Williams et al. In 1997, and protoplast fusion as per Pelletier et al. In 1995. Male sterility was artificially induced through mutagenesis also as per Call 1988. Features of male sterility. Male sterility prevents self-pollination and permits cross-pollination. Male sterility is an important outbreeding device which prevents autogamy, means self-pollination, and permits allogamy, means cross-pollination. Male sterility leads heterozygosity in a species as it promotes outbreeding and reduces homozygosity due to elimination of inbreeding. In male sterility, female gametes function normally. Male sterility results from the action of nuclear genes or cytoplasmic genes or both. Male sterility caused due to pollen or anther abortion, while female gametes are functioning normally. Male sterility is assayed through staining techniques. Male sporophytes and gametophytes are less protected from environment than ovule and embryo sac. In nature, male sterility occurs due to spontaneous mutations, as well as it can be induced artificially also by chemical or physical mutagens. Now, creation of male sterility. Male sterility can be induced or created by spontaneous mutations. Interspecific hybridization means use of different species during hybridization also results in male sterility. Mutation induction, genetic engineering, and chemically induced male sterility. Significance of male sterility in plant breeding. Hybrid seed production requires a female plant in which no viable gametes are born. Emasculation is done to make a plant devoid of pollen so that it is made female. In hybrid seed production of crops like rice, pearl millet, sorghum, mustard, and this castor, we are using male sterile lines where the female is sterile and the aerial pollination is taking place. In case of maize, where we are not using male sterile lines, so in such case we have to go for detasseling of male tassels in female lines. In other crops like cotton and tomato, okra, we have to go for emasculation process to make female sterile. So male sterility is very important aspect by which we can reduce the cost of hybrid seed production. Genetic male sterility is used in hybrid seed production, but it has limitations due to the need to maintain female parent lines as heterozygotes and segregation of fertile and sterile plants each generation. So in genetic male sterility, we have to go for rogging process where we have to remove 50% of fertile plants. Classification of male sterility. As per Call 1988, male sterility has been classified in 3 groups. First one is phenotypic male sterility, which is morphological based. Second is genotypic male sterility, which is governed by nuclear genes and cytoplasmic genes. And third one, chemically induced male sterility, which is induced by the use of chemicals. Under phenotypic male sterility, the male sterility is of 3 types: pollen male sterility, structural or staminal male sterility, and functional male sterility. Under genotypic male sterility, there are different types of male sterility. Like first one, genetic male sterility, which is also known as GMS lines, which is governed by nuclear genes. Second is cytoplasmic male sterility system, CMS lines, which is governed by cytoplasmic genes. Third one is cytoplasmic genetic male sterility system, CGMS, which is controlled by both nuclear and cytoplasmic genes. Fourth one is transgenic male sterility system, TMS lines, which is induced by the techniques of genetic engineering. Fifth one is environmental sensitive male sterility, which is divided in 2 types. First one is thermosensitive genetic male sterility, and second, photoperiodic sensitive genetic male sterility. And sixth type of male sterility is environmental non-sensitive male sterility. And third type of male sterility is chemically induced male sterility, where different type of chemicals are used to create a sterility system. Now we will go through phenotypic male sterility. Phenotypic male sterility is based on morphological or phenotypic expressions which represent the absence of male sex organs, lack of normal anther sac, inability of the pollen to mature, and inability to develop normal pollen, because of which the pollen will be non-functional and it will not fertilize the female stigma. There are 3 types of sterility based on phenotypic characteristic. Like first one is pollen sterility, in which male sterile individuals differ from normal only in the absence or extreme scarcity of functional pollen grains. This is the most common and the only one that has been playing a major role in plant breeding. Second is structural or staminal male sterility, in which male flowers or stamens are malformed and non-functional or completely absent. Third one is functional male sterility, in which perfectly good and viable pollen is trapped in indehiscent anther and thus prevented from functioning. Means in this type of sterility, the pollen grains are functional, but there is no dehiscence of anther, no bursting of anther sac, because of which these are not mating with female stigma. Genetic male sterility system (GMS). The pollen sterility, which is caused by nuclear genes, is termed as genic or genetic male sterility system. It is usually governed by single recessive gene, a small ms or s with monogenic inheritance. A male sterile lines may be maintained by crossing it with heterozygous male fertile plants. Such a mating produces 1:1 male sterile and male fertile plants. In case of genetic male sterility system, if we are going for hybrid seed production, in female line, which is GMS line, we have to go for roguing of 50% of the population, because these lines are producing ratio of 1:1 sterile and fertile plants. So we have to remove fertile plants and then it should be crossed with male plant. Now application of genetic male sterility in plant breeding. Genetic male sterility is applicable in production of hybrids in both vegetatively propagated crops and crops important for fruits or seed yield. The genetic male sterility system is used in wheat, maize, barley, sorghum, lucerne, cotton, sunflower, tomato and cucurbits hybrid seed production. And the advantages of male sterility can be described. GMS is usually recessive and monogenic. Hence, fertility restoration in the hybrid and the crossing plants are relatively easy. GMS can be used for the production of hybrid seed in both seed propagated crops and vegetatively propagated species. Genetic male sterility system requires less area and labor because the breeder has to maintain only A and B lines. A and B lines are having the difference only of fertility and sterility. B lines is used to multiply A line. Now the disadvantages. The genetic male sterility system is less stable. Sometimes GMS line can be fertile at low temperature. Like example, in rice, the sterile plants become fertile at temperature below 23 degrees centigrade. The sterility, which is influenced by the temperature, is called as thermosensitive male sterility. Cytoplasmic male sterility, CMS lines. Cytoplasmic male sterility is determined by the cytoplasmic genes, which is mitochondrial or chloroplast genes. Result of mutation in mitochondrial genome mtDNA is known as mitochondrial dysfunction. Progenies would always be made sterile since the cytoplasm comes primarily from female gametes, and the nuclear genotype of male sterile line is almost identical to that of the recurrent pollinator strain. Male fertile line, which is also known as maintainer line or B line, is used to maintain the male sterile line A line. Cytoplasmic male sterility is not influenced by environmental factors like genetic male sterility, so it is stable. And the CMS male sterility system show non-Mendelian inheritance with male sterility inherited maternally. In general, there are two types of cytoplasm in cytoplasmic male sterility. N, which is normal, and second aberrant, S, which is sterile cytoplasms, and these types exhibit reciprocal differences. Cytoplasmic male sterility is utilized for producing hybrid seed in certain ornamental species or in a species where vegetative part is used as an economic value. In those crops plants where seed is economic part, cytoplasmic male sterility is of no much more use because hybrid progeny would have chances of male sterility development. Cytoplasmic male sterility is not influenced by environmental factors such as low or high temperature. In other words, the sterility is stable. This type of sterility is used in crops like onion, fodder jowar, and cabbage seed production. CMS cytoplasmic male sterility is governed by plasma genes. Therefore, it is impossible to restore fertility in the hybrid. So, cytoplasmic male sterility is not playing major role in hybrid seed production. Limitations of cytoplasmic male sterility system. There is undesirable effect of cytoplasmic nature of male sterility system which is causing unsatisfactory fertility restoration. There is unsatisfactory pollination. A spontaneous reversion is taking place. There is nature of modifying genes because of which in case of hybrids the male sterility is also prevalent. There is a contribution of cytoplasm by male gametes, and these are having the less impact of environmental effects like low temperature or high temperature, so stability is there for sterility, and there is no availability of suitable restorer lines. So these are the limitations of cytoplasmic male sterility. Cytoplasmic genetic male sterility, CGMS. This is also known as nucleoplasmic male sterility and governed by both nuclear and cytoplasmic genes. When nuclear restoration of fertility gene is known for a cytoplasmic male sterile system in any crop, it is called as cytoplasmic genetic male sterility. The sterility is manifested by the influence of both nuclear and cytoplasmic genes, means with Mendelian inheritance and maternally inherited genes. The fertility restorer gene is generally dominant and is found in certain strains of the species or may be transferred from related species. So in case of cytoplasmic genetic male sterility, we are using the efficient restorer line. Since this gene restorer male sterility in the male sterile line is known as restorer line, this system is known as cotton, maize, jowar, bajra, sunflower, rice, wheat, etc. CGMS system is also known as three-line breeding system. In this system, three lines are used. First one is cytoplasmic genetic male sterile lines, which is known as A line or female line. Second is maintainer line, B line, and third is restorer line, R line. B line is identical to the A line in respect of male sterility. A line is sterile while B line is fertile. So it is used for the multiplication of A line. In hybrid seed production, A line is crossed with R line, restorer line. As R line, restorer line possesses fertility restorer gene in dominant condition, though the cytoplasm contributed by A line is sterility inducing one. As a resultant, F1 hybrid, which is used as a commercial hybrid seed, is fertile. A line and B line are exactly same genetically, but they differ only in respect of cytoplasm content. A line is sterile while B line is fertility having. A line has sterility inducing enzymes, whereas B line has normal cytoplasm. The R line possesses fertility restorer genes in dominant condition, and R line will be always fertile with respect to the cytoplasm, either it is sterile or fertile. And this is one of the method which is used for hybrid seed production of most of the crops like cotton, pearl millet, sorghum, flour, hybrid rice, etc. Now transgenic male sterility system. When the male sterility is induced by the techniques of genetic engineering, it is called a transgenic male sterility system. It is heritable and basically comes under genetic male sterility system. There are two kinds of gene involved in this. One gene which causes male sterility is integrated with genome A line, while the other suppresses it, which is R line. Recombinant DNA techniques is used. which is for disturbing any or number of developmental steps required for production of functional pollen within the microspore or for the development of any somatic tissues supporting the microspores. Transgenes for male hostility are dominant to fertility. Also, to develop effective fertility restoration system for hybrid seed production, transgenic male system is used, and some examples like Barnage or bar star system of hybrid seed production in mustard, which is related to GM crop. Now, environmental sensitive male hostility system, which is of two types. First one is temperature sensitive male hostility. In this type of male hostility system, male hostility is produced by the recessive MS gene, male sterile gene, at higher temperature like 23 degrees centigrade or higher temperature in case of rice. Temperature below critical point, there is normal fertility, and this male hostility is generally used to develop hybrid rice in China. And there are two lines like UPRI 95-140, which is temperature sensitive male hostility, and UPRI 95-167, which have been isolated as spontaneous mutations and registered as germplasm for hybrid seed production activities. Second type of environmental sensitive male hostility is photoperiodic sensitive genetic male hostility. In this type of hostility system, the expression of male hostility gene is drastically affected by the prevailing photoperiod, which is provided with the temperature within critical range 23 to 29 degrees centigrade for rice. Within this temperature range, complete hostility can be obtained for rice under long day condition, where the minimum duration should be 13 hours 45 minutes. Generally, short day condition obtain normal fertility in case of photoperiodic sensitive genetic male hostility. Now, chemical induced male hostility. The chemical which induces male hostility artificially is called as male gametocide, and this is the rapid method, but the hostility is non-heritable. In this system, A line, B line and R lines are not maintained. Some of the male gametocides used are gibberellic acids in rice and maize crops, sodium methyl arsenate in rice, and malic hydrazide in case of wheat and onion crops, which will cause male hostility. Here to go through limitations in using male sterile line. It is very difficult to identify the male sterile line when it is controlled by nuclear genes before anthesis. Male hostility cannot be effectively used in those crops in which efficient natural pollen dispersal mechanism is lacking and flower morphology is not suitable for cross-pollination. The existence and maintenance of A line, B line and R line is laborious and difficult and also costly. In exotic lines, these are not suitable to our conditions. The native and adaptive lines have to be converted into male sterile lines. Adequate cross-pollination should be there in between A and R lines for good seed set. So there should be selection of restorer line for getting the good yields and quality during hybridization. Synchronization of flowering should be there in A and R line for getting the good yields and good seed set compatibility. In some cases, genetic male sterile lines becomes fertile under low or high temperature condition or certain change of environmental conditions. Fertility restoration should be complete, otherwise the F1 hybrid seed will be sterile, and isolation is needed for maintenance of parental lines and for producing hybrid seeds. So this presentation was all about male hostility in plant breeding and use in seed production. Hope this will be useful to all of you. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Srivastava. Having request, please visit the channel, subscribe it and provide your kind and valuable feedback. Thank you.